This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 205, recorded on September 5th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining us today from Small Things Considered, Elio Schaechter.
Well, hello there.
Hello again. Is everything well with you?
Everything's fine. Thank you.
Very good. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
And from a hurricane-swept Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And it sounds so quiet there, Michael.
Right now. Yeah, we're in a lull. The house was shaking just before you called.
Yikes.
You can look out the window and see trees moving back and forth.
Oh, yeah. Bend
Moving
sideways.
is probably an understatement, huh?
Yeah.
You have palm trees down there?
We do indeed. And they have lost some fronds.
Yeah.
And the bald cypress is losing its leaves faster than I'm losing hair. So it's really something.
Could have been worse. It could have hit land near you, right? That would have been bad.
Yeah, if the eye wall came ashore, we were going to see 120 mile an hour winds. And we were lucky in that the gusts were only into the 75s and occasionally a 90. But it's pretty sporty. I live on a river. I live on a river, and you can actually go surfing in the river. And they showed a video from the College of Charleston. One of the students was out there soaking wet, skateboarding, except he wasn't pushing. The wind was just pushing him on his board, and he was skating all over Charleston on his board. And they had, one of the TV guys out there watching him push the board.
What river are you on, Michael?
The Ashley.
Ashley.
There's two rivers that form the Charleston Peninsula. The Ashley is, if you're looking directly down, is on the left-hand side, and the Cooper, or the Koopa, is on the right-hand side. And together, they form the Atlantic Ocean, or according to the local.
So how far up the Ashley are you located?
I'm about 3 miles up the road, so to speak, from the mouth of the Charleston Harbor.
Huh. Mm. So you're... I see. So there's North Charleston. You're not that far back, right?
I'm not. I live off of Hampton Park, which is the home of where the Formosan termites invaded the United States in the 1900s. They came ashore on a box of wood from Formosa that was a packing crate, and they took up residence in Hampton Park, which is one of our great parks that was designed by the famous architect Burnham, the same one who designed Grant Park in Chicago, where the famous egg is. And so Burnham designed it, except the termites have, the Formosan termites, that's where the founder effect is. So we're forever.
I see you here. I've got you on the map. You're not far from downtown.
No.
Very nice. And you're next to the Citadel. Huh.
Yes.
Do you have hurricane shutters?
No, I don't. I don't.
So I'm going to the whole podcast. I'm going to be worried about some branch come flying through and shattering glass.
We have no trees. We have no trees that can hit, and the windows are hurricane rated to 150 miles an hour. So they will actually take the impact. It's one of the code requirements. Like Florida, after Hurricane Hugo 30 years ago, the city went crazy with building codes, making certain that the houses would be more survivable and wouldn't cost people a lot of money to repair subsequent to catastrophic storm. And so
it's safety.
They have done it, and Clemson has a building program where they specifically look at tying down houses, tying floors together. the hurricane straps, and all sorts of things are codified and engineered into the building. But that doesn't always mean the builders actually follow the code. I mean, what I've been fighting today is I think they have neglected to put flashing around the window, because some of the windows that don't open are actually taking in water inside the house. And I think they forgot to flash the windows, and the wind is driving the water into the house.
Well, we're glad you're okay.
Yeah, so am I.
And
I was looking forward to doing this because anything but the hurricane.
That's right. Well, thanks for
Plus,
joining us.
the paper is really cool.
Oh, no, the paper. Anytime you have Asgards, it's very
If you like the work we do here on TWiM, consider supporting us financially. You can go to microbe.tv/contribute. We'll have more on that later. We're going to start you out with a little bit of a different snippet today from Elio.
Is it ever different? We're being very cosmic. We're talking about little creatures called tardigrades. I'll tell you what they are in a minute. But first, I'd like to tell you something that many readers probably caught in the papers. Namely, a few months ago, an Israeli private company sent a lunar lander to the moon, and it crashed. And the reason why this is interesting is because in the lunar lander were these tardigrades, these little creatures. And I'd really like to tell you about it. But first of all, why did they do that? Well, the founder of the foundation that sponsored this wants to, quote, create a backup of planet Earth. In other words, since we're about to destroy our planet, we might as well figure out how to live in other planets. And one step in that direction, maybe a far-fetched step, is to send little creatures to the moon. So, what are tardigrades? Tardigrades are little, they're also known as water bears or moss piglets. Tiny little creatures. They're about less than a millimeter long. In other words, they can be seen with the naked eye, but barely. And the interesting thing is that they are able to resist all kinds of horrors, like drying, high temperatures, low temperatures, radiation, etc. In fact, they had been taken out of a spacecraft on the moon and exposed to outer space. And they were retrieved, and guess what? They made it. They crawled away once rehydrated. So what are tardigrades? Oh, by the way, we may discuss this a little bit later. Was that a good idea to send these creatures to the moon? And well, it turns out it's not illegal. There are rules that NASA has, which says that if it's likely that a biological entity sent out in space interferes with the study of life, then it's a bad idea. But the moon is not a place where life is likely to happen because it's the moon. so it's very hot during the day, very cold at night, and very lacking in water. Anyhow, but just to put you at ease, this is not the only living thing on the moon. The Apollo astronauts who went to the moon left about 100 bags of human feces. So there's plenty of life there. So anyhow, tardigrades.
Are we supposed to be reassured by that, Elio?
Well, you didn't want them to bring it back because, the poor guy in this, the command module, he'd all of a sudden get a whole bunch of new stuff coming with him.
Yeah, you're right. It's questionable. Anyhow, there's life on the moon, but it's not going anywhere. Tardigrades. It's a phylum of water-dwelling microanimals. They have 8 legs, and they look like little bears, if anything. They are found all over the place. They're found on lichens and mosses, but they're also found on sand dunes, on soil, on leaf litter, and in marine and freshwater sediments. And there are many hundreds, if not more, species of these guys. They look like, as I say, like bears, but they have 4 stubby legs. They have a body cavity, which is a hemocell. They have no respiratory organs, so gas exchange occurs across the entire body. They have a mouth, no eyes. They use the mouth. The mouth is a little like teeth, which are used to pierce plant cells or small invertebrates. They are boys and girls tardigrades, and the females are usually larger and more common. And they are oviparous, that is, they lay eggs which are usually fertilized externally. The eggs hatch in less than 14 days, and the babies come to life with a full complement of adult cells. They go back in fossils to the Cretaceous. They live for up to 2 years, depending on the species. But the characteristic thing is they're capable of suspending their metabolism. This is known as cryptobiosis. And while in that state, the metabolism lowers to less than 0.1% of normal, and the water content drops to about 1% of normal. They can go without food or water for more than 30 years. And when you do that, then you then rehydrate them, they simply crawl away and start foraging and reproducing. But the amazing thing is the physical conditions they can withstand. They can withstand, for instance, a few minutes at 300 degrees Fahrenheit, 151 degrees centigrade. They can stand a few days at minus 200 degrees centigrade. And they can stand 30 years at minus 20. That's centigrade, which is freezer temperature.
So how does the water not disrupt their cellular structure? How does it not evaporate? And
I'm glad you asked. The water apparently evaporates, but what they have is they have a large amount of a sugar called trehalose, which protects the membranes.
Oh.
But in addition, they have a unique type of disordered protein, not the kind of protein that has a regular shape, but it's disordered. It's a class of proteins that we should sometimes discuss because they're really fascinating. And they serve a similar purpose of protecting it because the protein replaces the water in the cells and adopts what is known as a glassy, vitrified state. In other words, as the animals dry out, they become glass-like. That's the best answer I can give you. This is a subject of some study, and I can't tell you much more about it. They also resist radiation. Radiation up to 1,000 times the amount of radiation that would kill other animals. So they're amazingly resistant. The resistance to radiation has to do with another protein, a DNA-associated protein, which is unique to tardigrades and which suppresses DNA breaks by radiation. So it improves tolerance to radiation by suppressing the X-ray-induced damage. However, they are not totally resistant. That is, even though the resistance to radiation is amazing, it's not total. So the idea is that on the moon, if these guys are just simply, if they were spit out of the capsule and lying on the surface, they would probably die. However, if they were covered by a debris of the spacecraft or something like that, they may survive for quite a while. I'll point out to you that there's no plan right now to go and look on the moon and see whether they're alive or not. So we just have to take it on faith. So it turns out that one of our friends on this podcast, Mark Martin, who teaches at Puget Sound University, is a great fan of tardigrades. And if you want to see much more about it, you can go and look in his on his website.
I was actually wondering if we should start a Kickstarter campaign to send Mark Martin to the moon to rescue these stranded.
He would love it, believe me.
He would go. He would definitely go. He would contribute to the Kickstarter campaign immediately.
Or he'd at least be the lead fundraiser for the campaign. Yeah.
That's a good idea. Anyhow, the issue of whether it's a good idea to send living things out in space is debatable, to say the least. But so far, there's no real reason to worry too much about sending the tardigrades. But I must say, it's not clear to me why they did it, because even though this idea of learning about possibilities for backup to the planet Earth, not having planned for a retrieval mechanism to find out if, in fact, the tardigrades survive or not, makes the experiment rather incomplete. What do you think?
Yeah, I mean, and they were the only species that were sent, right? It's not like it was Noah's Ark or anything.
No. Well, they did send a whole amount of genetic information in very special metal plates. So that's the amount of information that's available there for the next person who goes to the moon, coming from some other planet maybe. There's a lot of stuff there.
They sent 100 million human, plant, and microorganism cells.
Yeah.
And did they send just the DNA code, or did they actually send DNA itself?
It says
the code. It's the code, I think.
Just the written code.
Yeah.
It says here the cells too. They sent in tubes.
They sent cells
sealed in epoxy with 100 million human, plant, and microorganism cells. So so unless they're in UV resistant containers, they're going to be the DNA is all going to be destroyed, right?
Mutated. My goodness.
I don't think they intended for their spacecraft to crash. And
no, they didn't.
and so that presents a big problem with the living stuff that they sent, because the crash probably destroyed all of the materials, because we have no way of knowing it. Since this is the 50th anniversary of Armstrong and Aldrin's first trip to the moon, there's been a lot of coverage on their first adventure to the moon. And one of the things is that at the same time they were landing on the moon, those of us who were around remember that the Russians were at the same time, or the Soviets were landing an unmanned spacecraft on the moon, and they had intended for that spacecraft to collect samples and then blast off and beat the astronauts back to the Earth with the first moon rocks. But unfortunately, that spacecraft crashed just like the Israeli one and never made it back to Earth. And Aldrin and Armstrong were the first two to bring back the samples from the moon.
When we did our TWiM with Mark Martin, Michael, you may remember Mark had a couple of toy tardigrades on his microphone.
Yeah, he prints them.
He has a 3D printer.
He has a 3D printer.
Yeah.
And he really encourages his undergraduates who take his intro micro class to really marvel at the wonder of the microbial world. And these are truly wondrous beings that, they're not prokaryotes. They are God-fearing eukaryotes. And
they're hardy microbes, in fact.
Yes.
Yeah. Would you call them microbes? I don't think I would.
Well, you can't see them unless you have a lens. I mean, I can't see.
Well, you can see a half a millimeter.
Yeah.
It'd be a little tiny dot.
I used to be able to. Not anymore. I now need glasses.
I don't know if you remember, a couple of years ago, supposedly the genome of a tardigrade was sequenced, but and they were surprised that a huge amount had seemed to have come from other organisms, but it turned out it was wrong.
No.
It was contamination, unfortunately.
Oh.
And I haven't heard since. So
if you're interested in Mark Martin's really creative ways to share microbiology, you can follow him on Twitter.
Oh, yes.
You can at Mark Martin.
Mark Owen Martin, I think it is.
Yes, and of course you can go back to TWiM 201 to actually hear him talking about, as Michael would say, the wonders of microbiology.
Hey, we had a great time talking with him about how the papers that he uses to introduce microbiology as a discipline to an undergraduate community. And they're not papers to trivialize. I mean, these are hardcore papers that graduate students often struggle with, and he exposes the undergraduate community to them, and he argues that they're ready for them. You just have to walk them through it, and they get it.
Yep.
And they marvel at it. So it was a lot of fun. If you haven't listened to TWiM 201, I encourage you to listen how Vincent and I had a good time with Mark.
And that's a great segue to what others have already called a paper that's going to get right into the textbooks that Vincent's going to cover.
Yes, our paper for today, thank you, Elio, by the way, for the tardigrades.
Of course.
Our paper was suggested by Ben, who wrote to the tetracoccal TWiM team. That's right, we're tetracocci, aren't we?
We are.
4 of us. I'm sure you've already heard of it, but I think this new paper on bioarchive would be great for TWiM. Briefly, the authors culture the Asgard archaeon Loki and propose a new theory for how archaea related to this one could have given rise to eukaryotic cells. The work of Woese becomes increasingly important as the years pass by. Would love to hear a few more archaea papers on TWiM because I think I am in a similar boat to many microbiologists where the extent of their undergraduate archaea teaching was that they're extremophiles that gave us TAC, which we know to be supremely understating their importance. I remind you, Ben, that TAC is from the bacterium Thermus aquaticus. It's not an archaeon.
So when I went to graduate school, we learned about this microbe called Metabolism omlianskii. It was a consortia that was isolated out of Ralph Wolf's lab, and Wolf's lab was down the hall from Woese. And the story goes is that this was a consortia of strain S and strain S and strain MOH that coexisted. And unfortunately, it was lost to time because they failed to carry it forward to the next generation, if you will. And what it was a really clever mixture. And if you think about what we're going to talk about today, here the S strain and the MOH strain oxidized ethanol into acetate and methane. The S strain of this consortium ate the ethanol, making as its waste 2 moles of acetate and 4 moles of hydrogen. And strain MOH then took the 4 hydrogens and fixed CO2 to make methane. But as I said, unfortunately, the consortium was lost to time. And since then, there have been a large number of syntrophic isolations. And so Vincent's going to take us on this journey of syntrophy. And so I think this is really the remarkable aspect of this paper is it reintroduces the concept of syntrophy to the audience. And we don't often talk much about syntrophy, and yet we take advantage of it.
Our listeners will be very happy to hear what it means. How about it, Vincent? Do you want to tell us?
Syntrophy, one species living off the products of another species. It comes that definition from all small things considered, in fact.
Oh, my God.
Did you know you had a definition on your blog?
Yeah, actually I did, yes.
Yeah, that's why he was encouraging us to get to the point.
So this is a bioarchive preprint, which is presumably in review somewhere, and it will be published somewhere very soon, most likely. Or who knows, they may make them do another 12 years worth of experiments. So the title of the paper is Isolation of Archaeon at the Prokaryote Eukaryote Interface. Is from a group from a number of institutes in Japan. First 2 authors, Hiroyuki Imachi and Masaru Nobu. And the last author is Ken Takai. And as the title says, they think they have a very early organism isolated at this interface. So let me tell you a little bit of background about this. This is figuring out the origin of eukaryotes. As Eugene Koonin says, it's a fundamental and forbidding evolutionary puzzle. Where did eukaryotes come from? And to give you the background for this paper, back in 2010, a gravity core was taken in the Rift Valley in the Arctic Ocean near a hydrothermal vent site called Loki's Castle. Of course, Loki was a Norse god, as you know.
For mischief.
For mischief. And they did metagenomic sequencing of this core. They didn't isolate any organisms. But based on the sequences, they found a small number. So these are archaea sequences, and they saw a small number that were clearly similar to eukaryotic proteins. And so things like cell membrane deformation, shell shape formation, cytoskeleton, properties of eukaryotes seem to be encoded in the genome of these archaea. And so they called this Loki archaeum. And of course, they didn't have an isolate. They didn't have an organism. They just had sequences. But comparing these sequences to all the other sequences we have, looked like both the Loki archaea and eukaryotes had a common ancestor. And so that's one of the words you'll hear a lot today, Loki archaeum. And another one you'll hear is Asgard or Asgard archaeota. This is a proposed superphylum which would contain the Loki archaeota, the Thor archaeota, the Odin archaeota, and the Heimdall archaeota.
No, there's a pattern there.
I guess
you sort of get the theme.
They're all Norse gods, right?
Absolutely.
So there's some evidence that eukaryotes arose from an ancient archaea. And the idea is that the mitochondria came from a bacterium that was a symbiont in that. And as I said, they have only this sequence that leads to this hypothesis. It's very powerful. But in this paper, they believe they've isolated one of these organisms. So let me tell you how they did that.
This is not for the faint of heart.
This is just remarkable.
Be committed.
This is remarkable. So they have uncultivated deep marine sediments and the microorganisms. They set up a methane-fed continuous flow bioreactor to keep these deep sediments alive. And they ran this for over 2,000 days. The idea being they would enrich for anaerobic organisms that would live down there in the methane seep sediments. And so they feel by sequencing this, they enriched many diverse microbes, including Asgard archaea members like the Loki, etc. Then they inoculated a sample of this bioreactor community in glass tubes with simple basal media, and they waited a year.
But you got to remind the audience that they put into their medium 4 bacterial suppressing antimicrobials: ampicillin, kanamycin, streptomycin, and vancomycin,
right? Because
those are the antibiotics that are specific to prokaryotes, both the cell wall and their protein synthesis.
Right. And the medium was mainly amino acids. These antibiotics, they incubated them at 20 degrees. And after a year, they said, we found faint cell turbidity. Can you imagine? Who would have known? So they did some sequences, and then lo and behold, a very simple community was present. A small population of Loki archaeota, these resembling the sequences that were found down at that hydrothermal vent. Some halo desulfyl vibrio. So they called this strain MKD1. They subcultured it, they did more PCR, and they got it to grow a little bit more. And listen to these growth properties. You inoculate new cultures with these microbes. Lag phase of 30 to 60 days before they start dividing, 3 months to reach full growth, and they get about 100,000 copies of ribosomal RNA per milliliter. So the doubling time is somewhere between 14 and 25 days. Very slow growing.
So much for the advantage of working with bacteria and archaea.
That's right.
They double fast.
Keeps the competitors at bay, though.
It does.
I just, how you get this funded, you got to be doing something else in this on the side, right?
You forgot to mention their affiliation. They're from the Institute for Extra Cutting Edge Science and Technology of Avant-Garde Research of the Japan Agency for Marine Earth Science and Technology. We need one of those institutes here in the States.
We do.
Yeah, and then they collaborated with several other institutes in Japan. But yeah, you're right, Michael. It's specially designed to go after the really deep and big
profound questions.
Yeah.
All
right. So this culture that they end up growing, it has 3 microbes. They call it a triculture. It has Halo desulfyl vibrio, about 85% of the culture is that. Methanogenium, 2%, and their MKD1. And they look at this under the electron microscope. You can see these organisms are physically closely associated. They did the genome sequence. So now we have an archaeon, a Loki archaea, and then we can get the entire genome from one microorganism. And so they can see what kinds of metabolic activities it has. And through some experiments, they found that MKD1 can actually degrade amino acids and peptides together with the 2 other components of this triculture. The MKD1 degrade the amino acids, the hydrogen or formate goes to the other microorganisms, which then take it up and use it. So this is, as they say, has been a 12-year journey. They actually were able to eliminate the halo desulfyl vibrio, and they end up with a pure culture of the archaeon and methanogenium. And it was a 12-year journey to get this, and the name they propose is Candidatus prometheoarchaeum syntrophicum strain MKD1. Okay, so now let me tell you a little bit about this strain. The cells are very small cocci, generally forming aggregates. They seem to have some extracellular polysaccharide-like materials, no visible organelles within the cells, which of course is very different from eukaryotes. But they make these very interesting membrane vesicles and chains of blebs on the outside. So these are very interesting protrusions. And in fact, since this is a bioarchive paper, anybody can go see it. You can see the pictures of these very unique protrusions, and they'll become important later on. They did analysis of the lipid composition, and as you know, you can distinguish archaea from bacteria based on the lipid composition, and these MKD1 and the methanogenium revealed typical archaeal signatures in their lipids. They confirmed that this MKD1 can degrade amino acids anaerobically. They fed them labeled amino acids. They produce hydrogen and formate, and these are picked up by the other cohabiting species. They tried lots of different culture compositions and never got better than giving them amino acids or peptides. So then looking a little bit closer at the genes, MKD1 clearly has genes for degrading 10 different amino acids and generating from that hydrogen and formate to get rid of the electrons. And those are picked up by the 2 other microbes that are growing with this. These pathways can recover energy by degrading these amino acids through a oxoacid intermediate. It's an acid with an oxygen group like pyruvate. And it can degrade them either hydrolytically or oxidatively. And that gives us acyl-CoA, and that can be further processed for generating energy in the form of ATP.
And if I could just interject, for those of you following at home, figure 2A is a really nice layout of the metabolic pathways that Vincent is referring to. So you can see how this all comes together and the partnership that develops between these three different cells.
Yeah, so the partnership is key because the partners take the hydrogen or the formate that this MKD1 is producing by degrading amino acids. And they are making, in turn, compounds that the MKD1 can use as well as themselves.
And they have to continuously be in close proximity to each other because it's as if you or I are dropping $100 bills in the airport. Mm-hmm. If you're not next to somebody you know who will return your money, someone else is going to pick up that energy. And, these things are in very close proximity, and the EMs really show you how close they really are.
Mm-hmm.
And they make an extracellular matrix that helps hold the consortium together, which is
That's
brilliant.
Right.
So none of the $100 bills will escape to anyone else.
So this partnership can be involving SMKD1, can involve sulfate-reducing bacteria or methanogenic archaea. And they do some experiments which show that the MKD1 can switch between hydrolysis or oxidation of the amino acids depending on what partner is present. So if only one partner is present, it will do one thing, and if the other is present, it will do the other. It's very interesting. They look at the ribosomal RNA sequence. They do a phylogenetic tree. You can see clearly that this archaea MKD1 is clearly a sister with the eukaryotes. So they say it's the closest cultured archaeal relative of eukaryotes. We've never had one cultured. It's amazing, right?
Yeah, it's figure 4. It's really remarkable.
Then they look at some other archaeal sequences, and they can see some of these enzymes that they've been looking at. And they think that this hydrogen-evolving amino acid degradation, the use of partners, is probably a common feature across the Asgard archaea members with all of those Norse gods' names that we talked about earlier.
Pay special attention to the genes that light up for electron metabolism, because that's the key to how this consortium is making its living, so to speak.
Right.
And they do a beautiful job of summarizing this in their figure 4B. They've got a really great graphic that shows how each of the members of this, what did you say, family?
The superphylum.
The Norse gods, the superphylum. The particular contributors to amino acid degradation, electron metabolism, et cetera. So really great job of summarizing a lot of data.
So MKD1, amino acid catabolism, hydrogen metabolism, and lack of biosynthetic pathways, which I didn't mention, but very few biosynthetic pathways. They propose that the last common ancestor, the last Asgard archaea common ancestor to eukaryotes was just like this one, amino acid degrading anaerobe producing hydrogen and fatty acids as byproducts, and it acquired ATP from phosphorylation of 2-oxo acid intermediates and depended on partners. In this case, one or two other partners. So from these data, they have a new eukaryogenesis model, which they give the wonderful name entangle, engulf, enslave model for the origin of eukaryotes. And here's how this is really wonderful how it goes. So the idea is the ancestral Asgard archaeon was a archaea very much like MKD1. It was a syntrophic organotroph, and this is now in Earth's early oceans. Its partners were probably methanogenic archaea, not sulfate-reducing bacteria, as they pulled out of contemporary sediments, because the sediments today are very different from the sediments 2.7 billion years ago. So back then there was very low ocean sulfate concentrations before the Great Oxidation Event, which happened about 2.7 billion years ago as the ocean and the atmosphere became oxygenated, mainly as the result of oxygen-producing cyanobacteria?
Because we ran out of hydrogen sulfide in the ocean, and the organisms had to figure out where else to get their electrons from, and they figured out, well, we'll get them from water. And we can then capture photons and be able to make our energy that way.
So after this great oxidation event, the ocean, the atmosphere becomes oxygenated, the marine sulfate concentrations rise, and so then they say, okay, maybe the MKD1-like organisms then began to interact with sulfate-reducing bacteria and get their energy that way. In the process of this great oxidation event, you get more organic matter transferred to the marine sediments, so it's becoming a very rich place in which to live. And then they say this ancient Asgard archaea could have gone one of two ways to survive and to adapt to these new conditions. They could have stayed in the mud, in the anaerobic habitats at the bottom of the ocean, or they could move towards the layer of oxygen. And of course, there would be more substrate and more energy availability. And so they think that last archaea before the eukaryote division probably grew close to the sulfate and organic rich interface environments.
Remember, the great bottleneck in all of metabolism is where to throw your waste. So if you can throw your waste into a molecule that's just out there, like oxygen, and you can throw your waste electrons into it, and the only byproduct that you get from that is water, which is relatively innocuous to the living cell, you're then given so much more competitive advantage than the poor anaerobes that have to put their waste electrons either into organic acid, or they have to do tremendous gymnastics to throw their electrons into things like methane, where they fix CO2 and put it into methane. So this is a selective advantage for this organism to be able to dump its electrons into a molecule like oxygen.
So, Michael, I thought you just dump your waste on the moon.
Well, that was the last paper.
Oh, sorry.
That was the last paper.
So you're getting more and more oxygen, right? How do we adapt to this? How do we use it, as Michael says, as a terminal electron acceptor? And they say, okay, two things could happen. These organisms, remember these ancient Loki, archaea, and their relatives, they could acquire all the genes you need for aerobic respiration, like the whole electron transport chain, the terminal oxidases, or they can get an endosymbiont that already can utilize oxygen to get rid of waste. And so they think the first thing actually happened in the least the ancestral Asgard archaea probably took the former route. They acquired the genes for using oxygen, and the archaea closer to eukaryotes took the latter route, acquired an endosymbiont. So we have a three-member interaction. We have sulfate-reducing bacteria. They can scavenge hydrogen from the archaeon and the aerobic partner. And so these three are together. They're growing well. The archaeon doesn't have a lot of biosynthetic pathways. One of these partners was probably the alpha proteobacterium that became mitochondria after it was taken up into the archaea. And this evolved over time. It led to some transitional states and eventually endosymbiosis of the alpha proteobacteria into the archaea.
Yeah, genetic reduction.
Right. Now, how did this happen? This is a very interesting thought process here. The presumption is that the archaeon engulfed the metabolic partner, right? But they say phagocytosis is energy costly, and there probably wasn't enough energy in this tiny archaeon, right? Remember these tiny cocci, and they're too small to engulf the partner. And these archaea don't have the phagocytic machinery. We don't see it encoded in their genome. So then they say these protrusions, maybe these trapped the partner, the alpha proteobacterial partner that's using the oxygen. And eventually these all fused to produce a cell containing what became the mitochondria.
This is really the a crucial point of the paper, isn't it? Because this is really totally original. As far as I know, nobody has ever proposed something like this.
I think Baum and Baum proposed the inside-out model of evolution of eukaryotes, where these membrane protrusions coming from the archaea themselves fuse and then trap another microbial cell within.
Right.
Who did this, did you say?
Baum and Baum.
Baum and Baum, 2014.
There's a reference in reference number 38, right? So here we have now an endosymbiosed alpha proteobacteria inside an archaea. And these, under aerobic conditions, these shared amino acids, derived oxoids needed for hydrogen. But when exposed to a little bit of oxygen, they could use oxygen to accept electrons as the waste products, as Michael said. So as this evolved, having this endosymbiont was good. It would produce a lot of energy for biosynthesis, and so the endosymbiont was maintained. But of course, you have to streamline the process, as Michael hinted at before. You get rid of genes that you don't need anymore. You get rid of the lipids that you don't need anymore. So they lost this ancestor lost the archaeal type lipids and ended up with what we see in eukaryotes today.
And some horizontal gene exchange.
Now, a key point here, a major point in making this symbiosis work, was the evolution of an ATP transporter, right? And they say, interestingly, it's unrealistic to think that ATP-providing machinery evolved altruistically. And so they think that the alpha proteobacteria actually developed these as a parasitic tool to absorb ATP produced by hosts. And in turn, so now you imagine the bacterium is inside of the archaea. It's just sucking up ATP. Stealing it. And so the host then evolves not to produce it and generate an ATP concentration from the endosymbiont to the cytosol so it can steal it back, reverse the direction of ATP flow. And eventually that took advantage of these transporters. And so that took away the parasitic aspect of the relationship. And so they say we arrive at this last ancestor. It's got symbiotic arrangement here, and it looks very much like today's eukaryotes. So a lot of that is speculating, of course, and they say we need to get more of these organisms to study them and figure out if this is right.
Yeah, I was really thoroughly enjoyed just how provocative the paper was and how much of their paper they spent really speculating and building models from their data. And it occurred to me that they were waiting years to get their system up and running and to start collecting data. So they probably, they naturally slowed down and had time to really think deeply
That's right.
about the science. So it's really a piece of scholarship. Beautiful.
Yeah, I just am impressed that someone would persist so long, right? Because, they waited a year just to get a little bit of turbidity in those cultures. It's amazing.
And they didn't throw them away.
It's remarkable. It really is.
I'm sure they had other things going on.
Of course. So in the end, we now have a living microbe which is ancient in terms of very close to, it's not the actual eukaryote, of course, because it's got separate endosymbionts, not an endosymbiont, but it's got separate bacteria that it associates with. But their properties leads them to develop this hypothesis of how that could have led to the endosymbiosis. And they've covered a lot of the alternative things that could have happened, and I think it's really convincing. So I would just check it out, because even if you just read the discussion, and the figures, as Michele said, are great. It's open access. I'm not sure I did such good justice to it, but the figures are wonderful.
For a virologist, I thought you did great.
Thank you. Thank you very much.
We're not going to ask you to memorize the pathway like we all had to do when we took physiology in the ancient times.
I think the pathways are fascinating. They are.
And the educators out there can use this to really hook students and convince them to really learn the metabolic pathways. Plus, as I said, the graphics are terrific, including their final figure, which lays out this evolutionary pathway that they are proposing.
What I think is very cool, Michele, is that this pathway figure 2 is wonderful. It shows you the linkage between these 3 bacteria. Mm-hmm. It's all based on a little bit of experiments, but they look at the genes and they can predict what's going on. Infer, right? Exactly.
Yeah, based on a wealth of physiological and metabolic data that's already available,
of course,
from other systems. Yeah, so this was an effort by 22 different people, but the two co-first authors, one is Hiro Amachi. He's a senior scientist at Japan Agency for Marine Earth Science and Technology. He was born in Sunan, Yamagachu. He earned his PhD in engineering at the Nagawaka University of Technology. Pardon my pronunciation. And that's where he became fascinated with cultivating of fastidious and symbiotic organisms using engineered ecosystems. So while studying these bioreactors, he found that they contained very high diversity of organisms that nobody had been able to culture yet. So he was inspired by this and decided to tackle the problem in reverse. He wanted to engineer bioreactors that would allow him to enrich these microbes. So he then moved to the Japan Agency for Marine Earth Science and Technology, where he was a research scientist, and that's where he built this reactor that allowed them to culture organisms from the deep sea marine sediments, which was known to be rich in uncultured organisms. So by setting up this one-of-a-kind, lab-accessible ecosystem, over the past decade, Hiro has been able to cultivate now a wide diversity of these novel organisms. He partnered with Masaru Nobu, who also goes by Nobu, who's a research scientist at Advanced Industrial Science and Technology in Japan. Nobu is a Japanese American. He was born and raised in Fairfax, Virginia, and he studied biology and geology in Carrollton College, which is in Minnesota. He then went to the University of Illinois, Champaign-Urbana, and got his PhD in environmental engineering, where he was applying genomics and bioinformatics to study microbial symbioses and the metabolisms of these organisms that we hadn't yet figured out how to culture. And his motivation was to figure out how we could convert waste into energy that we could harvest. So he then began his work at the Advanced Industrial Science and Technology Institute as a research scientist and began to broaden his focus to other ecosystems and continued to use genomes as blueprints for each organism's way of life and look at the genes to get clues to their evolutionary history. The two of them teamed up and developed their own symbiosis, with Hiro doing the cultivation and Nobu the genomics. And together they produced this beautiful work. Now, when they're not being scholars and scientists, Hiro enjoys cultivating not only fastidious organisms, but also raising 3 kids. Who themselves can be a bit fastidious, and he is blessed to have just a wonderful wife to share that journey. And then Nobu, his current hobbies are cooking and fish coral keeping, and he's also starting a new life chapter in Japan with his wife. So that is their story, and obviously they formed a terrific symbiosis, a great partnership that's better than either one could have done on their own with their own specialized traits.
Impressive.
Great story.
Yeah.
So this paper was posted on bioRxiv on August 6th. So, could be out in a month, could be out in a year. Who knows what the journals are going to make them do, but
it's interesting. Science, the journal Science, which is a premier journal, did a feature on their bioRxiv.
That's right.
Preprint. So I wonder what's happening.
Yeah.
Well, Science would probably truncate the wonder of this paper way down, and you may not get all the richness that you have in the bioRxiv paper.
Yeah.
I mean, Science and Nature have a tendency to do short things, and you lose a lot of the detail that is really quite convincing in their EEE model of entangle, engulf, and enslave. I mean, that is going to be the hard one to convince reviewers, because it is speculation until they get more data, but the data take 12 years to develop. We're not going to wait another 12 years.
What one person calls speculation, another person might call model building, and it's really based on quite a lot of research that they cite. So I think that's real scholarship, synthesizing what is out there and putting it into a testable model. So, of course, it is going to take time, but hopefully the next one won't take 12 years, huh?
I'm looking forward to seeing what this paper looks like when it's published in a journal, and we'll let you know when it comes out. We have two. Thank you, Michele, for doing that. Mm-hmm. We have two short emails. Let's read those. One is from Shannon, who writes, "Hello, TWiM folks. In regards to the last episode 203 on the lack of mechanistic research behind probiotics, would you all be able to conjure up some educated insight as to the safety of these spore-forming bacteria for the use of probiotic supplementation / gut health? For example, Dr. Amy Myers sells a product geared toward SIBO, small intestinal bacterial overgrowth, containing the following." And she sent the supplement label from the bottle. One capsule contains 3 billion cells of Bacillus subtilis, Bacillus clausii, and Bacillus coagulans, with the note that the daily value has not been established.
That's true.
I've read some Bacillus are resistant to some antibiotics and could transfer that to other bacteria in the gut. What are your thoughts on colonizing the gut with a spore-forming bacteria? Thank you, Shannon, an avid listener of TWiM and all its related podcasts. Any thoughts on this?
We already have Bacillus in our guts, so they're already there.
And I'm not sure these would colonize. They could certainly pass through, but whether they colonize or not is a separate question.
Yeah. I think that this is an area of growing interest, obviously, and I found a wonderful summary article called "Bacillus as Potential Probiotics, Status, Concerns, and Future Perspectives," which basically goes into all this in great detail, Shannon. It says basically you have to evaluate each strain on its own, and it has to be tested for safety, obviously. But yes, as Michael said, a lot of our food contains Bacillus that we eat on a daily basis. And yes, animals are fed Bacillus, and they contain antibiotic resistance genes. So obviously, you would like to avoid that. But I don't think spore forming per se is a problem, right?
No.
And my understanding is there is data on the transit time for Bacillus subtilis through the human gut.
Mm-hmm.
I think it's on the order of, I want to say 8 hours, something like that. So it passes through, but doesn't stay and permanently colonize.
And the trick with the strains that are listed is really whatever trait the microbe may be displaying in the human gut condition. And when I was working on the microbes associated with the inoculum of whole plant corn silage, this was a product that was produced by a major seed company to encourage farmers to use the inoculum so they would get more weight gain per unit of food fed ruminants. We learned that even though the organisms were all the same genus and species, there were subtle differences in the strains, even though they were, Bacillus subtilis. And she lists this as Bacillus subtilis HU58, and it's trademarked, whatever that means. But it really is what trait HU58 is producing, whether it's a small molecule that could be influencing the quorum sensing going on in the gut. There's a lot of communication going on, and even though they may not colonize, they may send a signal for your normal flora to do good things. And this is, of course, the emerging area of research, and we did a lot of work once upon a time trying to figure out how do you make a good inoculum so that you can make the best forage available to put weight onto animals. And it's a complex problem that requires a lot of careful work, many, many controls.
Taking weight off of humans would be even better.
Yes. If you do that, you retire. You retire.
So Shannon, the article, I'll put a link in the show notes. It's open access, so you can check it out. We have an email from Reed who writes, Dear Scientists of TWiM, my name is Reed. I recently finished my undergraduate degree in biology at North Dakota State University, working full-time in a pancreatic cancer research lab for a year until I go off to grad school. In the past year, I have had a slowly culturing interest in microbiology, seriously considering going to grad school to earn a PhD in microbiology, but I'm looking for places to start. In my search, I discovered TWiM and was delighted at the content and discussions that were brought up. I have started binge listening at episode 1, and after 15 hours of listening, still have about 8 years of science until I'm caught up. That being said, I apologize if my question had been answered in one of the 180-some podcasts that I'm waiting to listen to. In my biology undergraduate, much of what I learned about microbes and their study had been disease-focused, and much of the microbiology that I know involves study of disease. While I have utmost respect for this, it is not a huge interest to me, and I believe it has given microbes a very bad reputation that is completely undeserved. What I believe is fascinating is the use of microbes in ways that can benefit everybody at once, and not just a small portion of the population that may be afflicted with a given disease being studied. Topics such as microbes for the use of energy, such as electrogenic or methanogenic bacteria, use of microbes to clean environmental pollutants such as plastics and oil spills, and the use of microbes to increase the well-being of already fairly healthy individuals absolutely fascinates me. Do any of you scientists at TWiM have suggestions of topics for me and others interested in the use of microbes for utilitarian purposes to look into, including expansions in the one I've listed? Are there any research groups that you know of doing this kind of work and would recommend to a potential graduate student? Any help greatly appreciated. Thanks for the great podcast. P.S. When saying the word human, many of the scientists on the show pronounce it as human, whereas I have always heard the pronunciation human. Is this just regional differences as I am from North Dakota, or have I learned a mispronunciation?
I don't remember saying human.
I think we all
human
say,
and human sound very much alike.
Yeah, I think we all say human, right?
I think so.
Yeah. But
anyhow,
other people say
advice about where to go to school.
Yeah, where should what should he do? And these are interesting interests, of course. And probiotics is another one, right, that we talked about just before.
Mm-hmm. Probiotics is definitely going to be the growth issue. It's really about funding today, and unfortunately, we're funding infectious disease research, and how microbes interact in the treatment of cancer seems to be very popular. And
how about phages?
Phages have never, while they're the darling of science fiction, they don't garner much research funding.
Plus, it's mainly for therapy, right? And he doesn't want to do
infections.
He doesn't want to treat infections.
I would look for labs that are funded by the National Science Foundation, labs funded by the Department of Energy. But unfortunately, those labs are being zeroed out because it's getting harder and harder to fund investigator-initiated science without an immediate outcome. It's all this translational research that everybody is very much interested in funding today, rather than the paper like we just discussed, which really requires forward thinking on the behalf of governments to say, well, what might come out of this?
But having said that, there is a lot of fascinating research going on in the fields that you mentioned, Reed. I also can direct you to a report from the Academy of Microbiology called Microbe-Powered Jobs. You can find it on the World Wide Web. It'll be free through the asm.org website. And it can give you some more ideas. But if you're interested in how beneficial microbes can protect our health, there's certainly a great group here at the University of Michigan, led by Pat Schloss and Vince Young and others who are doing some really cutting-edge work in that area. But, there's also really neat lab at Michigan State looking at bioenergy, Hemorrhagera. We could go on and on.
Would it be good advice for him to go to ASM Microbe next year and get a sampling of all the stuff that's going on?
It might be overwhelming.
Yeah.
It would certainly be a pleasure, but it'd be like the biggest smorgasbord you've ever been to.
But you'd certainly
so you'd have to plan well. Yeah,
you'd get everything, right? All these areas are represented. Yeah.
I would actually encourage anyone to look for strong microbiology programs. So not think about one particular lab I want to be in, but great programs. And we could name our favorites, but probably that wouldn't be appropriate.
My favorite microbiology program is Elio Schaechter's.
Yeah.
Hey. Wow.
And Small Things Considered is a good place to start because you pick a lot of really cool things that fits his menu items.
Well, thank you very much. I like the sound of that.
I agree with Michele. For your PhD, you need to just do good microbiology, right?
Yeah.
Right.
And for your career, that determined later.
Yeah. But Reed realized that many strong PhD programs, not only do students not pay tuition, but they also get a very generous stipend and health benefits. So it can be as much as $30,000 that you are paid each year to be a PhD student. So go for it.
Okay. That is TWiM 205. You can find it over at asm.org/TWiM. And if you listen on your phone using a podcast player, please subscribe so we can tell how many people are listening. If you really like what we do, you can support us financially over at microbe.tv/contribute. You could, for example, sign up at Patreon or PayPal to give us $1 a month or whatever you choose. It would help us with our expenses. And finally, if you have questions or comments, you want some advice, TWiM at microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter's at Small Things Considered, which is a wonderful blog you should check out. Thank you, Elio.
Thank you.
And Michael Schmidt is at the Hurricane University of South Carolina.
This is true today.
Thank
yes,
you, Michael.
Indeed.
But you're past the danger zone, right?
Yeah, the storm has moved up. We're just now continuing with the rain. The house is no longer shaking, thank goodness.
Very good. Thanks for joining us, Michael. Appreciate it. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.